Hello everyone, how are you today? Not so good? Good. So, today we are getting started a new chapter, which is about now DNA interactions. So, to have an overview about this modern genetics and molecular biology, but first off, we need to take a look at a brief history through which we are here nowadays. Okay, so this genetics began with the so-called classical genetics, which is also called the transmission genetics. Okay, and made a gradual, smooth transition into modern so-called molecular genetics, like this. However, still, like in school, if not anything else, school, you as a student of life science, student are to take both the classical genetics course as well as the molecular genetics course in the form of several different forms, like including molecular biology and some other newer branches of this relevant entities. However, if you really think about deep, then these so-called classical genetics and molecular genetics cannot be separated. Actually, it's one thing, one just continuous branch of life science, but simply the way people view their perspective and the way they try to solve the problem in the subject has been a little bit changed. Okay, although still, for the sake of convenience matter, still we do have the separate subject as if they are totally two different courses, but in reality that is not. Okay, so what actually this classical genetics has provided us was this concept of genes. Okay, so through studying this classical genetics, we get to understand the actual what genes are and how they behave, and how can we predict their behavior along with the activity of heredity. Okay. And then when we made a transition into so-called this molecular genetics, then now people try to find out the actual what's inside, the little things, actually the molecular or chemical nature of these genes and how they behave at the level of these molecules. Okay. So that's what we are going to take a look at in this chapter. Of course, the level of what we are surveying in this chemical nature of genes and their behavior would be only the surface scanning type of very fundamental, basic level though. Okay. So anyway, the Catholic priest Gregor Mendel, Father Mendel, actually was the founder of this classical genetics. As I say, the classical genetics, another name for classical genetics is a transmission genetics, because it explains the heredity, the way the genes are being transmitted from one generation to the next and next generation. That's why it's called transmission genetics. It's also known as transmission genetics. That's the classical genetics. And what he did was, so he lived somewhere in the 19th century in Austria. So his contribution was the correct concept of gene, what exactly genes are. And he made a simple but thorough observation, after observation over many generations of this garden pea plant in his monastery, and he recorded and analyzed the pattern of the inheritance, segregation of these sort of the traits that everyone knows vaguely at least what traits are, right? And then he correctly understood the concept of genes, because at the time, at his contemporaries, peoples, the general view of genes was something like vague, and it's something like, oh, you have your father's genes and you have your mother's genes, and as a as a child, you get half of the father's gene and half of the mother's gene, again mixed, just like they think this gene as if some kind of a liquid. So you have fathers, we can compare this father's gene as a blue liquid and mother's gene as the red liquid. And then if you mix these things, then your liquid, the color of your liquid will become the purple, like something. That's kind of, although funny it may sound, but that was a mainstream idea about the genes at the time, the 19th centuries. But so he made an observation. What's going on? Hey. Always here and there, several different kind of problem always. So this is what he observed. So he chose actually initially 7 different characters. So here, using this figure, I'd like to actually have an opportunity to look at and try to distinguish the difference between character and trait. Although I mentioned the difference between these two terms earlier in the beginning. So characters are over here in this column, the character column. You can look at the different types of characters. Flower color is one such character. Flower positions, seed color, blah blah blah. And human height, human eye color, human intelligence. All of them could be different characters. Okay. So, in other words, if I am allowed to explain the nature of the characters in a modern term, then I can easily explain that, okay, those characters are something that is coming from your different genes. Different genes is responsible for different type of characters. Okay. But what about the trait? Traits are under specific character. There can be different alternative. Here, in his example, in Mendel's example, I don't know, it's just almost like a miracle. But then he somehow he chose the characters in garden pea that displays only A or B types of alternative characters. Okay. That's possible, but there may not be such A and B. There are only 2 alternative different versions of traits, so-called, under single character. If you think about it, there are so many different types of traits under single character that displays several different types of alternatives. But somehow, he was able to avoid such confusion. And miraculously, he chose those characters that shows only A or B type of 2 alternative. That was really the key to the success of his experiments, though. But anyway, he did that. So in this flower color character, there were only purple or white, two different colors available, which we categorize or define as two different traits under the character of a flower color. Okay? Like if you think about it, the human height as a character, how many different traits can you find in a human trait? Countless different traits exist. But so if he somehow accidentally got involved in such thing and then tried to follow things, then he may not have been able to come up with this simple and accurate and precise law of the segregation, Mendel's, the famous laws. But somehow he was able to avoid, but simply chose these alternative characters like this, and then crossed these two parents. Okay. And then surprise, 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 only in the next generation, every offspring's, children's, were all displayed only either one of those two parental traits. There was no such a thing like a mixture, as I briefly mentioned that, okay, father's gene, mother's gene, you as a child will get the half the characteristic. Nothing, none of them. Only, so something to his eyes at this generation, at that stage, it appeared that these two traits, one of these two traits has just gone, was missing. Where did it go? If he stopped at this point, then he wasn't able to. He wouldn't have been able to actually discover this law of genetics. But he became at least, he decided to become persistent, and then he did again, okay, using all this first generation in is self-fertilized, then to see if actually one of these traits has disappeared, or next generation somehow it may have a blessing of miracles to reappear, and then it did. Okay. So he kept this thing and then analyzed, thought about it, what is going on, and then came to a conclusion that, okay, so from this observation, this is what I think that the characteristic was the nature of the gene. Okay. So first off, this gene is not something that mixable. The problem of this mixable theory of a gene is that once these two genes, parental two genes are mixed, and there is no way you isolate this thing into individual thing. If those genes are something like liquid-like thing can be mixed. Okay. That's actually, there is a particular terminology, but for such theory of a mixable gene, it's called the blending. Oh, please. Yes? There are, okay, I know. There are some of those things, exceptions to this Mendel's law, okay? If I'm going to go over those different exceptions and explain what was there actually the real reason, probably, yeah, it's not really convenient. So let's just skip out of those things. I will explain, if you are interested, I will explain those things after class. Can I? Those exceptions. There are certain exceptions. Some of them are actually the result of the environmental influence, and some of them are actually result of the epigenetics, and blah, blah, blah. Yeah, but here, let me just clarify the basic framework of the Mendel's principle of genetics first. Okay? So those are called blending hypotheses. Those are the leading idea about the genes at the time. Blending hypothesis is just like liquid. You mix these two different colors of liquid and then you present some halfway around the color. However, if that was the case, then fine, you get the half the characteristic, but what's next? There's no way you actually re-isolate. What do you need to? Why do you need to re-isolate? Actually, yeah, you don't really have any particular occasion to have such idea about Mendel did. And then, on the contrary of this leading idea, his idea was genes as this discrete particular nature, something like this stick. Genes are something like this stick. They can, and another thing is pairs. Every individual, regardless of humans, animals, or plants, each organism can carry two, not a single, but two genes per each characteristics. Okay? Then what happened is these two combined and then separate throughout this reproduction. These two genes can temporarily be separated and then recombine, okay, during the course of the reproduction. What comes to your mind if he explained, like if we can somehow bring Mendel to this classroom and then actually ask him to explain what he thought about the genes are like this? And what reminds you the most? Doesn't it something like I think I have seen it, something very similar somewhere, because especially if these genes are something that highly closely related with this sexual reproductions, right? Then the version that he explained as a gene, the nature of gene, is strikingly resembles something that we know already, which is what? Genes exist pairs. And then during sexual reproduction, these pairs get separated. And then during this reproduction, which probably through fertilization, they reunite and then restore this pairness. Haven't you seen this thing before somewhere else? In this class? No? Haven't? Totally unheard of this? What are you talking about? Isn't it something like if I gave you a kind of clue, what? Something like you originally exist as a diploid, and during sexual reproduction you temporarily become haploid, right? Which as a gamete or egg, okay? A gamete or egg, sperm or egg, and then reunite once you become a fertilized egg, you recover this diploid status. What am I talking about? This ploidy chromosome number, isn't it? So he was viewing this gene as exactly something that of the behavior of a chromosome during the sexual reproduction. Through meiosis, you exist as a diploid cell, right? Diploid, which means each chromosome you have double pairs. And through meiosis, you generate temporarily haploid cell in the form of egg or sperm, depending on who you are. And in sperm cell or egg cell, the ploidy is a haploid, which means only you have a single chromosome, one chromosome. Okay. Genes are on chromosome. Whatever they are, genes are on the chromosome. So if he was following those characters, which are present on different chromosomes of garden pea, and scored and analyzed and to understand what genes are. Then that's how he exactly came up with this. Okay, so they behave like this, but to our eyes, to modern humans' eyes, that was exactly the behavior of this chromosome: segregation through meiosis and fertilization. Okay, and remember, as long as you remember that genes are, whatever they are, genes are on the chromosome. Okay, so that's exactly what he, without knowing that exactly what they are, that's what he was talking about. Okay. So this chromosome, just like the chromosome, these genes as a particular, okay, discrete unit can be separated and then reunited, recombined during the course of this inheritance. You mean the sexual reproduction. Okay. So then if that was explained actually. Only this type of mystery can only be explained only if he assumed that this trait exists in pairs, okay? And then got separated and recombined in the next generation. And here there was some kind of hierarchy. So, for example, so now then inevitably we need to introduce these two different terminology about the status of this gene. Okay. If those genes, okay, genes are responsible for different traits, right? If these genes in a single individual exist, okay, in pairs, but the nature of these genes are exactly identical, then we call those status as a homozygous. Okay, same. You have two genes for each character. You have two genes, but if those two genes are 100% identical, then you call them as a homozygous. That's an adjective. Okay, adjective. And homozygote is a noun. But if hetero always means that different. Heterozygote means these two genes that you have is a slightly, no matter how slight it is, but they are different. But then you call them as a heterozygous. That's what this sentence tries to explain. Then in the homozygous status, there is no problem, no conflict, because they are just like same identical twin-like things. Each genes, there should be any kind of a conflict. What kind of a trait should I represent? There is no such trouble. But there is potential trouble. What if these two genes are different in terms of a trait that the gene is making, like eye color? Okay, you have two genes that is responsible for your eye color, and one is responsible for black eye, the other one is responsible for brown eye. Okay, so that is exactly heterozygous. Then what eye color would you or should you have to have? Mixture of black and brown, or black or brown, or what? Your left eye is black and your right eye is brown? That's actually more convenient and exciting. But anyway, so in such heterozygous status, only not two, or not the mixture, but only one of the two, as he observed, and as most of you already understood in these days, only when there was two trait, purple versus white, but at least in the first generation, all of the offspring shows of the purple, which means the white color trait is gone, but it's not actually permanently gone, but simply disguised, masked. Okay. Why? Because white trait is recessive over. Recessive to, I shouldn't say over. And purple trait, purple color is dominant over the white color. So that's what we call casually in our everyday life, casually say something is dominant over the other one. Hey, this is dominant, okay? Over this kind of. You casually speak about some of the human related trait as, okay, some people even say short height is dominant over the tall height, which is incorrect. But anyway, people casually say those stuff like that. So that's the definition of dominant or recessiveness. So in a heterozygous state, only one of the two allele shows its character as a trait. Then we call that as a dominant allele. Dominant allele, the function is it shows its own characteristic trait and masks the other alternative trait. So masking the effect, the other one, which we call the recessive allele, is not showing phenotypically. Okay. So here is another term, phenotype. The opposite term of this phenotype would be genotype. Genotype is the actual makeup of the genes. But in many cases, it can be hidden. We cannot detect as an outside observer the genotype. Only what we can decipher, only we can detect is a phenotype that this certain trait from a certain character give us to you. Okay. So that's the phenotype. But just because this phenotype is a purple color, that doesn't mean that the genotype is something all identical. Two genes are purple. It may be heterozygote, like purple and white gene, but since the purple trait is dominant over white trait, so what you see from outside is only purple color. It's deceiving you. That's the nature of dominant versus recessiveness of the genes. It's all about. So that's, I forgot introducing this, something called the term allele. Allele is a synonymous, almost synonymous to trait, different trait, because for each character you have at least two different alleles. Why are there usually you have two alternatives? The definition of alleles is an alternative version, alternative form of a certain character is an allele or gene, alternative form of a trait or alternative form of a character. It's the definition of an allele. Korean name for allele is this. Usually, in many cases, actually, at least minimum, you have two different alleles you can discover per each different category or per each different character. Why? Why do you get to usually discover, get to have two different alleles? Why? Or should I rephrase this question? What is the maximum number of different alleles a person can have? What is the maximum number of different alleles a single individual, human, can have for each different character? How many? What's the maximum number? 5, 10? Two? Answer is 2. You can have only 2 different alleles as a single person. Can have only 2. That's the maximum number. You can have only 1 allele if you were homozygous. If you were a heterozygous, the maximum different allele that you can show is 2. Why? In other words, because you are a diploid individual. If you were tetraploid individual, the maximum number of different alleles would have been expanded to 4. The number of chromosomes will limit the number of different maximum different alleles that you can carry. However, if I make a different type of question regarding this allele, what is the maximum number of different alleles that can be possibly occur for a single character, a particular character? A given character can have how many maximum different alleles? What? 3? Why? Just guess? But the answer is this. Enormous, countless. Why? All these different alleles are the result of what? You have a single gene. Mutation, exactly. Mutation. Mutation will be able to generate different version of this gene. And how many different ways you can mutate this? In as many as you can think. Because mutations are by definition a sequence change of a gene. DNA sequence change is a mutation. So yeah, that's the two different kind of a perspective, okay, for these alleles. Single person can have only 2 maximum number of different alleles because you are a diploid. Okay. However, for a single gene, you can think of as many possible different forms because each different form of allele is a result of a mutation. Okay. So here, the follow-up stories of probably you have seen if you took high school biology, probably you got sick and tired of this Punnett squares and going through. Basically, I think this is a very silly way of. This isn't a math class, but you have to practice. You have to sharpen your math skill for these genetic problems. So I don't. I'm not really particularly so excited about this particular type of questions. But anyway, it simply explains what he meant because you have these 2. For each gene, the big word is represent the dominant allele. Small capital, small letter, little word, little alphabet represent the recessive allele. And since you're a diploid, you have 2 of these. So these parents were all homozygous. So if you cross these 2 homozygous individuals, every offspring, no matter how many you generate your children, every offspring will be heterozygous for that particular character. So this heterozygous status, as long as there is a dominance over recessiveness relationship exists, all you see is this one trait, which is a green color of this P. But if you become patient and then keep doing this, and the next generation, this allele will segregate once again, so individually giving out 2 different alternative forms. So now this phenotypically now the original parental phenotype reappear. And if you keep doing this, then yeah, you may be able to find a little more interesting observation later. Yeah, but anyway, that's not the scope of in this classroom. So simply try to explain the nature of the genes as Mendel thought. But to us, it sounds so simple because we already know the genes are always on the chromosome. So here on this, the right side figure is the rephrased version of this Mendel's the explanation about genes. Okay? He didn't know anything about the chromosome. So without knowing the presence of anything vaguely, the chromosome, that he still was able to precisely figure out correctly the nature of this gene was amazing. But to us, if we try to understand his version, okay, genes, we don't really have to think about what exactly genes are, but simply they are on chromosome, right? So the chromosomes are like tools of the location for all those different genes of your genes, everything are located on the chromosome. So each genes, like for this particular chromosome, if there were several genes are present on this particular chromosome, and each location of different genes we call as a locus. Okay, that's another terminology, locus. And loci is a plural. Many different multiples of location of the genes are called loci. Locus versus loci. So, for example, in these days, a geneticist casually refer the location of the genes in a particular chromosome. Okay, and chromosome number 13 at particular locus, there are certain genes, blah, blah, blah. And as we already know, there are a huge number of array of genes are present on each different chromosome, right? So that's that. If we pay attention to the other side of the story, like, so diploid organism, we are all diploid organisms, so this particular same locus, okay, this same locus is marked in color, but for some reason, intentionally, okay, the same locus was colored in two different colors. One is in orange, the other one is in yellow, but these both yellow and orange indicate the same locus, which means the same genes, because this is homologous chromosome. Okay? The thing that you are a diploid individual, but is because you have homologous pairs of chromosome for each different chromosome. So, on this particular locus, these genes, if these genes were the same, then this is homozygous. But as this color try to indicate, if these two genes are somehow a little different, then they are technically heterozygous. So now it becomes a matter of deciding who's win and who will lose in the form of this dominance over recessiveness. The one become expressed will become dominant, and the one remains silent, covered, not showing up, will become recessive. So only the one of these two alternative traits, only one dominant trait we can observe from as an outside observer. Okay? Although some occasions these two things behaves just like those blending theory, like some halfway around the mixture. So instead of like if you cross the red flower color and the white flower color to plants, and you anticipated that, okay, red is dominant over white, so all those opposite things should be the red. But some occasion, in some different types of species of plant, the flower color may exhibit the intermediate, halfway around, like pink color. Apparently, as if like trying to support the old-time incorrect theory of a blending hypothesis, but it is not actually. There is a reason why some occasions you get the so-called incomplete dominance. And I'm not going to explain over here because it's not really important for our purpose of this theme of the lecture. So only suffice to say that there are some exceptions. Always, you can find so many different exceptions to what the exact doctrine, so-called the doctrine of Mendel's doctrine. But just because there are several exceptions, that doesn't mean that Mendel was wrong. No, each different exceptions can be explained. Okay. So this is actually a more important question. So now, assuming that every one of us understood the difference between dominant and a recessive trait. Okay? But what if I gave you questions like, what is dominant or recessive trait? But simply, instead, if I gave you why is a particular trait is dominant, while the other one is recessive, what would you say? If I ask you what is dominant or recessive, then your answer should be dominant is the one that expressed or showing off in the state of heterozygous. In heterozygous state, the trait, an alternative trait, if this alternative trait shows off or expressed, then we call as a dominant. On the other hand, if this one is not expressed or not showing, then that is recessive. Okay, that's fine. That's the question of what they are. But if asked you why they are dominant or recessive, any idea? I said from a molecular genetics perspective. Anybody want to challenge? Yes. So you want 2 points? You already had 2. Okay. What do you mean second one? Why? Almost there, but it's not entirely correct. But you are halfway around, like, yes, exactly. You are getting there, right? Because you mentioned the functionality. Dominant one is dominant because it has full function. So if we try to explain the recessive trait first, why they are recessive, it's probably easier to understand, because the reason why the recessive trait become recessive is because throughout all of them are result of a mutation. Okay, some mutation will behave as a dominant, the other mutation will behave as a recessive. But also surprisingly, most of those mutations that you get to hear are mostly recessive. Okay? Why? Because those mutations are resulting in some loss of function. Original function is there. So unmutated one we usually call as an unmutated version of the gene as a wild type. Okay? So from a perspective of a wild type as a function, wild type, for example, like hemoglobin, is an essential. It provides an essential function for you to carry oxygen to every part of your different body, hemoglobin. Okay? But if you have a mutation in this hemoglobin gene, okay, and then if it results in some malfunctional, useless, not generating any active function, then it cannot behave as a real functional hemoglobin protein. You have just lost your function. A lot of mutations are just like that, because mutation occur randomly, and as a result, the wild type function has been lost. But now, in this status, if you had this wild type and mutant version of this hemoglobin at a heterozygous status, okay, which one would be dominant? Your wild type will be dominant because this one still can function. So this one has just lost, this mutant, recessive mutant has just lost the function and became a recessive one. Because as long as this so-called normal functioning hemoglobin is there, it has no problem of providing still this hemoglobin function. So the recessive one is not affecting any outcome because there is already some functioning alternative counterpart. As long as there is a functioning counterpart, it's recessive. The loss of a function doesn't have to even exist. You can go home because I can do alone by myself. And that's the nature of this recessive gene. Recessive gene is simply because it cannot provide any function afterwards. So you are simply just a non-existent. So whenever there is some alternative counterpart is partnered with, the one who can generate the function will dominate you. You shut up because you are useless. You are obsolete. That's the nature of dominant over recessiveness. Functioning one will become dominant over non-functioning one.
교수님,
Yeah?
근데 그런데도 그럼 아까 적혈구 이상, 결명 적혈구 비대증과 같은 그 대부분의 적혈구 이상이라는 열정인데 그런 헌팅턴 유형이나 ADPKD 같은
That's a dominant one. Yeah.
ADPKD 같은 그러한 계속 유전 질환이 있는데 그런 유전 질환은 왜 존재하나요?
Let me explain. Can I explain? So, then on the other hand, why certain mutation behave as a dominant? Guess what? This one, as a result of the mutation, is although rare, this mutant obtained a new function. If this one obtained a new function, the existing one cannot do anything about this. Because as a new function, it will present itself, showing off. Now, so that kind of although rare than recessive mutant, this dominant mutation, the reason why certain mutations behave as dominant is because simply because they somehow obtained a new function. But that's not all. There is another way of creating a dominant mutation, which is, can you guess? So dominant mutation will either the one which generate a new function so that this existing function cannot block or cannot mask anything about, or there is another way of creating this dominant mutation, which is even if there is an existing one, if this mutant somehow behave blocking, interfering this existing function, then it can become a dominant one too. It's all about the function. I hope you can understand this. It's all about the function of the gene. Then we simply classify accordingly. Okay, if you can find a new function or something that the existing function is somehow altered, interfered, then we call that as this is a dominant one. If this mutation cannot do anything, so in that context, she is right. In this mutation cannot do anything alone, okay? Because it simply just lost its original function, so that it can be complemented. It can be covered by the normal function, existing normal function, in the form of heterozygous, then it should be simply recessive one. So that's the functionality-based distinction of dominant and recessiveness. Okay? So, despite this, people didn't give a damn about what he has. He just actually published the paper. No one read. No one read, and no one actually agreed or recognized. Okay, you're right. So simply it went unnoticed until he actually passed away. Even something 30 or 40 years after his passing away, then now some independently, without even knowing Mendel's work, some of the independently working plant scientists, 3 different plant scientists discovered exactly the same thing of this Mendel's law. And only afterwards, then eventually later, people gave credit to Mendel's work as a original initial founder of this genetics. Okay? Because, understandably, it's exactly, if we knew about what chromosome and what's their function in terms of this carrier of the genes, then probably it would have been much easier for us to understand what Mendel explained. But at the time, nobody knew anything about this. Okay. So, yeah. So then, about 100 years after Mendel's first initial report about the nature of the genes, 100 years after, it's the early 20th century. It's not too long ago. Then only people realized, genes, those genes actually that Mendel talked about were actually on some weird structure like chromosome. That's actually a long story how people now finally recognize and admitted that, okay, this chromosome, that things, those weird structure located in nucleus are the one actually carrying the genes. Some intermediary supporting evidences and experiments. Okay. But the final nail on the coffin, final conclusion was made by the work of the famous, renowned Thomas Hunt Morgan, who was the expert in Drosophila, small insect, Drosophila small fly, and Drosophila expert. And accidentally, actually, he discovered the rare mutant in eye color. The Drosophila's eyes are all wild types. Eye color was all red, but white eye color eye mutant was discovered. And in pursuit of the origin, okay, how this, because he discovered some weird strangeness summarized in this diagram. Because even according to Mendel, back then everybody was adopting the Mendel's law of genetics. So to them, there was no problem of this having the standard genetic cross, Mendel's genetic cross, like using Punnett square or whatever. However, this occasion was something weird in that if you cross this female wild type and a male mutant, okay, then as you have expected, everyone in the first generation, everybody was showing the wild type red eye color. Okay, fine, there's no problem. But if you did this in the next generation, something weird thing in terms of this sexual ratio. Okay. So the mutant form reappeared, but only in males, and that is also only the half and half ratio. So to explain this, the only way to explain this is only because there are 4 chromosomes in Drosophila, and one of those four is a sex chromosome, like in X. Female have two X chromosomes. Male have one X chromosome and one Y chromosome. And like human, similar to human, the Y chromosome is physically quite different from X chromosome. So you can distinguish this Y chromosome. And it can only explain this strange outcome only if that mutant gene was located on the X chromosome. Okay. So the conclusion was so solid. So then finally, conclusion was, it was confirmed that that white mutant eye color gene was located on X chromosome. Then automatically that can be explained that all genes, if this mutant gene was located on the X chromosome, that means all other genes are on chromosomes. So that was the first incident that people now officially admitted or recognize, okay, wow, genes are on chromosome. Get it? Stupid now? 100 years after? Now you get it. Right. Fine. So everything was solved, it seems. But no, it's not. So genes are on chromosomes, then what's the matter? If genes are on chromosomes, then automatically DNA should the genetic material, isn't it? No, it wasn't that easy. So even after, okay, the genes are all located on the chromosome, fine. So everybody go home now, celebrate. So DNA as a genetic material, no. Wait a minute, why? Because still, majority of people, okay, wait, because something is chemically, DNA was already analyzed chemically. It's done. And it turns out that, chemically speaking, DNA is made up of half protein and half DNA. Do you still remember the famous proteins that present tightly associated with DNA? Histone, forming that nucleosome, right? Those histones, even if you weigh in terms of moles, half of those whole chromosomes are coming from proteins that histone, majority of them are histone, and half of them are coming from DNA. So which one is actually the genetic material? DNA or protein? What would you say? Without knowing any previous knowledge, if you were asked, given this data, okay, your chromosome consists of half protein and half DNA, which one do you think the genetic material? And the other one is providing some structural support. Almost everybody, the vast majority of the scientists back then pointed what? Proteins. Why? Okay, obviously, stupid. Protein should be the genetic material. Why? Guess what? DNA is made up of only 4 different types of nucleotide. A, G, C, T. Protein is made of 20 different types of amino acid. Genes are what? Genes should be able to provide diversity. Do you really think that only rotation of these 4 different kinds of nucleotides, do you think enough diversity can be made? Nobody. What about this 20? What's the combinatorial differences, like the different possibility? If you try to generate different type of genes through different combination of the 20 the different amino acids, huge. So that should be, protein should be obviously the genetic material, everybody, almost everybody. So the world renowned famous, at the time, even the Nobel Prize laureate thought protein should be the genetic material. But still some scientists thought otherwise. Okay, so here I should go over some of the two pioneering scientists who provide in terms of experimental evidence that, oh no, we think that DNA is actually, on the contrary, DNA is the genetic material. Actually, there are two lines of actual experiment, and the one first, it's not an experiment, but the first one is this simple observation to raise this question. I will just postpone this discussion till Wednesday, and I will just go back to your student questions. I really still think that going over this student question regularly should be a part of a regular lecture, because I really find so many different valuable questions, so that by just simply together going over some of the questions, we can have actually more benefit as a lecture. So let me announce how I want to go this shortly. Not today's, but today I will simply go over, but simply try to incorporate this thing into regular lecture thing soon, I guess. So the first question is, so what would happen? There was actually another very similar question about this. So what happens if the cytoskeleton actually is destroyed or malfunctioning, and what would be the outcome of this? It's really there are two different ways we can look at it. Cytoskeleton, of course, is extremely important. So if you have a malfunction in any of this cytoskeleton function due to some abnormal structure of the formation or destruction, then obviously all the function at the cellular level as well as the individual organism level huge effects. It will be heavily critically affected, because if you think about many different functions that we get from the cytoskeleton, including this cellular transport and cell division and movement through muscle contraction and even cellular level motility, you name it. So yes, however, on the other hand, the one that I brought up this question is another. The flip side of this coin is cytoskeleton is also deliberately, we call this a dynamic instability of this cytoskeleton. So we deliberately let this cytoskeleton structure is very unstable, so that easily broken out. Why? Because we have some intention. We have a secondary intention of, by easily breaking up this, then we convert this raw material in forming some other material. For example, like microtubule. Microtubules are mainly used as a main road for each different transport vesicles movement. Along those road of microtubule, these little transport vesicles walk around to deliver this each different protein sac or each different things. Okay, that's motor proteins are. But then the next another important function of the microtubule is what? Cell division. Once this cell come up with this cell division time, then you have to quickly generate the spindle fiber, which is also made of microtubule. So you have a choice. You resynthesize, you reinvent the wheel, you resynthesize from the beginning of individual all the spindle fiber that you need, or alternatively, you could break existing road of microtubule and get those individual subunit, and then reassemble quickly the spindle fiber. That's what the latter is true. That's what the cell does. So to do so, intentionally cells make all these different, each different cytoskeleton structure so fragile, easily breakable? Maybe that is the reason why there are so many, even unnecessarily, extensively, the network of cytoskeleton. If you saw, if you remember, as you saw the fluorescence micrograph of the previous lecture, that those different actin, microtubule, and inhibitory filament of the stainings everywhere. It's simply randomly trying to shoot in the dark instead of having some specific destination, but it's everywhere so that easily some of them can be broken and then reused in some other purposes. It's probably. The prokaryote not need any cytoskeleton. Yeah, obviously, apparently, prokaryote do not have any cytoskeleton. It's probably because they're small size. So it's just a matter of, it informs us that maybe such a small size is somehow some limitation of this, the efficiency of diffusion can reach. If you exceed this distance, then probably moving something through diffusion only is not efficient enough. And besides, as a prokaryote, you do not need to have any spindle fiber because you don't have any different chromosome to separate. So for such different reasons, prokaryote do not have to have cytoskeleton. This one, the question was asking how this person assumed that all those initial mutual interaction, mutual relationship begin as a potential parasitism, which is about probably right. Okay? If somebody tried to get inside of you, okay, not many occasions this person, the intruder, has a goodwill. Okay, I have this goodie, so let me present this good thing to you. So what do you have in return for me? Type of thing. Probably not. It's just simple, straightforward mind of exploitation. Oh, I want to take what you have. But probably somehow during the course of this adjustment, both parties somehow get to discover, realize that, oh, that guy has something very good that can be beneficial for me. And in return, I can give this to this guy. So this gradually develop into more beneficiary, because if there was some kind of relationship of a parasitic relationship, then this host will always try to destroy this visitor. Okay? But as soon as these both parties find the usefulness of each party, then it can last much longer and even forever, even in some cases. So that's why. So that's how probably transition. But it's all kind of speculation. No one knows. No one knows that what exactly happened. And the second question is, in the case of this mitochondrial symbiosis, why do these mitochondria still keep their own gene inside? Why didn't they just simply give them away to the nucleus? Some of the genes, it's much better for them to keep inside because the gene product, if this gene product is really larger, then as a mitochondria, if you gave this gene to the nucleus, the nucleus will make this gene expressed and then eventually finally make the final product, which is a protein. Then that protein have to come across, penetrate the two layers of mitochondria, if you think about. If that protein is too big, you have a problem. So usually there's a general trend that those still existing inside of the mitochondria, the genes are the one who make the big, larger protein which are being used inside of this mitochondria, the other one characteristically. So that can be explained. Okay. Is it technically possible to insert some of the genes into the chromosome? Like, for example, there are some of the essential genes that are responsible for making the cell as a stem cell. Yeah, we have discovered some of the key genes. And can we insert this gene into human chromosome so that transform this gene, the cell, into something like this stem cell-like thing? We have already, not we, people, those scientists who are studying so-called IPS, induced pluripotent stem cell. In Korean, it's called like almighty stem cell, it sounds like. It's called induced in Korea, in English it's called the induced. I stands for induced. Induced pluripotent stem cell. This pluripotent stem cell is like as if it's behaving something as if the embryonic stem cell, which is almighty, all capable of a stem cell. So there are technology already available. You can transform any normal cell, any regular cell, not stem cell, but simple regular body cell, and then convert. By simply modifying some of the genes, it is possible, it turns out that it's possible to convert this cell into stem cell. And those technologies are really promising because you don't have to touch the issue, the ethical issue. If you decide to stick with the embryonic stem cell research, then you have to deal with these ethical issues, even if you have a technically capable of. But in this occasion of induced pluripotent stem cell techniques, you don't have to worry about such things because you are converting any cell from your body, and then theoretically speaking, convert into this. So yes, answer is a yes, we can. Techniques are available, and then there is a way we can avoid even these ethical issues too. Well, I will just stop over here and visit with more questions ready next time. Okay, so today the lecture I will stop here.